Good afternoon, everyone. Thank you for coming. I'm delighted to give you an update on Gyroscope this afternoon. We are a retinal gene therapy company, so let me tell you a bit more about what we're up to. So our mission at Gyroscope is to preserve sight and fight the devastating impact of blindness. Our company was founded back in 2016 to explore the convergence of understanding of three areas of science. So we are focused on the complement system, which is part of the body's immune system. We also look at the genetics associated with retinal disease, and we also combine that with a deep understanding of gene therapy. And it's really only been in the last 5 to 7 years or so that data has become available across the range of those areas that enables the combination of complement genetics, especially in a large multifactorial indication like age-related macular degeneration, and gene therapy as a treatment modality to offer new therapeutic opportunities for these kind of diseases. So based on the science of our founding scientists, 2.5 years later, our lead program is now in clinic. So we started our first clinical trial earlier this year in dry age-related macular degeneration. We continued on our journey as a company earlier this year when we combined with a company called Orbit Biomedical. So Orbit Biomedical is a medical device business that's designing and developing a device and a procedure to deliver gene and cell therapy to the back of the eye. And that company now has become part of our team. And so we've now created the first fully integrated retinal gene therapy company that has clinical manufacturing and delivery capabilities. The company is headquartered in London in the UK, and we have a facility here in the US just outside of Philadelphia. And a couple of weeks ago, we announced the closing of our Series B. So that brings the funds raised and committed to date to the company to just over 100 million. So our mantra at Gyroscope is people, patients, and promise. So just to give you a bit of a sense of some of the people that are leading the drive that we're making towards the next wave of gene therapy. Our CEO, Karam Farouk, joined us earlier this year. So he joined us from Genentech, where he previously led the ophthalmology and immunology business unit, and he was responsible there for the Lucentis franchise in the US. Our Chief Scientific Officer, Jane Hughes, brings a real depth of gene therapy understanding and also a very broad understanding of the complement system, which she brings to us from MedImmune and from GSK. Mike Keane, who is our Chief Technology Officer, is a biomedical engineer. So his background is in device engineering and development. He's come to us from Janssen, and he was involved in designing and developing the Orbit subretinal delivery system, which I will tell you about shortly. Ashwin, our Chief People Officer, is helping us with our growth. We're expanding very quickly. So he brings expertise in growing companies and teams over the last 20 years from Juno and Grail and Genentech and others. And Ed, our Corporate Affairs Officer, brings us communication, but also patient advocacy experience, which is very important to us. So thinking a little bit about our patients and how we aim to help improve their daily lives. Age-related macular degeneration, or the severe form geographic atrophy, is a leading cause of vision loss. So there's an estimated 3.75 million or so people in Europe and US that are affected by late-stage AMD. And the incidence of disease is very much driven by demographics. So you can see on the right here, this is a chart just showing incidence by age. And so thinking about the aging of our population, this is a disease which is just going to grow. It's a long-term, progressive, slow degeneration of the cells in the macula, which is in the tissue at the back of the eye. And it results in a progressive and irreversible loss of vision, starting centrally and then progressing outward. So what you can see here in the picture on the left is just a depiction of what it looks like to an AMD patient, and that black spot in the center of vision just continues to grow over time. And it's hugely distressing for patients because they lose their ability to read, their ability to drive, their ability to live independently. And as you can see from the quote here, which comes from the Macular Society, which is a patient organisation in the UK, the worst thing is not being able to see faces anymore. The lady in the middle of the slide here is a lady called Janet Osborne, and she was the first patient treated earlier this year in our ongoing interventional trial. So she was treated in Oxford in a short surgical procedure, which was performed by Robert McLaren, who's a leading retinal surgeon. The procedure was covered by the BBC News, so if you're interested in it, you can Google it and find out a little more. So what is age-related macular degeneration? What causes it? Well, here there's a depiction of the progression of disease. So a healthy retina is shown here on the left, nice and clear. As AMD starts to develop, lipid deposits form under the retina, these little white spots, which are called drusen. And some patients at this stage with early AMD go on to develop neovascularization and form what's known as wet AMD. And this is treatable with anti-VEGF antibodies, such as Lucentis and Eylea. But there's an underlying disease progression that continues. So all patients progress on to the advanced form, which is called geographic atrophy. And you can see in the center of the retina there on the third picture along where the cells are starting to degenerate and vision is starting to be lost. And as it progresses, it covers the whole of the retina, results in blindness. So how are we thinking about addressing this indication, and especially with the gene therapy approach, given that most gene therapy programs to date are in rare monogenic diseases? So we're really trying to forge a path with making the benefit of gene therapy that has been seen across many other programs in many other diseases available to a very large patient population such as this in a large and complex and multifactorial disease. And the way that we're doing it is we're targeting the complement cascade. So the complement cascade is part of the immune system. It has a number of pathways and factors within it. And there's a multitude of data available in the literature that supports the involvement of complement, and in particular an overactivation of the complement cascade as being associated with the risk of developing dry AMD. So here's just some examples of some of that data. So for example, on the left, here you can see a picture of a drusen, which is that white lipid deposit that I showed you before under the retina. And what you're seeing here is the membrane attack complex, which is one of the final stages of the complement cascade, which is co-localizing with the drusen. And it's known that many complement factors are upregulated in AMD pathology. In addition, over the last few years, there have been a number of independent genome-wide association studies that have been looking at the genetics of AMD and looking at risk factors associated with development of disease. And what you can see here actually is the outcome of one of the studies, which looked at 30,000 subjects. And what they found was that there were a number of complement factors which had genetic mutations and were associated with an increased risk of developing disease. And increasingly, as people explore this space, there is emerging clinical data which demonstrates a role of complement. So here's just an example where an anti-complement approach was able to show slowing in the growth of the lesion size, so the degeneration of the cells on the retina. So this is all encouraging. It gives us a great sense that we're working in the right direction. And our approach really has been to take this and to focus in on a very defined subset of patients as being our first study where we look at our particular complement approach. So our lead program is GT005, started in the clinic earlier this year. We have a number of programs coming behind. So in addition, we plan to address a broader patient population within the GA landscape. So that program is currently preclinical and should be in the clinic next year. And we have a number of other programs behind that targeting other inflammatory retinal diseases. And the goal with our lead program, GT005, is to develop a one-time treatment which aims to slow or ideally halt progression of disease. So we're able to focus in on a very specific patient population because of data sets like this. So this was a study that was done looking at 3,500 subjects, normal subjects and AMD subjects. And it was looking at mutations within a particular factor within the complement cascade and classifying them based on in silico analysis into mutations that were predicted as being benign, possibly damaging, probably damaging, or loss of function mutations. And what you can see here is that the type of mutation is correlating with levels of that factor in serum. And there is a group of patients, which you can see that are highlighted here in yellow, that have probably damaging or null mutations that are associated with low levels of factor in serum. And also looking at the colors of the little diamonds there, these patients were much more likely to develop advanced disease. So we have taken this data set and other supporting data and focused our first clinical trial on exactly that. So it is actually called the FOCUS study. This is a phase 1/2 trial. It's running across 7 sites in the UK currently. It's a phase 1/2 open label dose escalation study. And we're looking at the safety and the biological activity of our approach. And we do it based on natural history data that we've been collecting across an AMD patient population. So we are identifying patients that have low serum levels of the factor that we're delivering and that also have high-risk genetic mutations. And this is a very specific patient population that we're targeting for our proof of concept. And this is a very elegant approach because it enables us to address a very large indication, a multifactorial disease with a very focused proof of concept. Our view is that because we believe AMD is associated with a general dysregulation or imbalance, if you like, of the complement system, that by showing a proof of concept in a focused patient population, we also believe that's a mechanism which is going to be applicable to a broader group. So behind the FOCUS study, we have a large natural history trial, which is called the SCOPE trial. This study is running across 36 sites in US and Europe, and it's gathering important data around disease progression in advanced AMD patients, but also looking at their genetics and looking at their complement type. One other thing that's really important to us is the importance of delivery. So gene and cell therapies, the vast majority of them are delivered into the back of the eye into something called the subretinal space. And the subretinal space is the right place to deliver to for these kinds of therapeutics because it's right in between the target cell types of the retinal pigment epithelium and the photoreceptor cells. And it's an enclosed space, and it's the space which has been associated with the best clinical outcomes for patients to date. And it's achieved by a short surgical procedure, which sounds like it should be easy, but you actually need to access that piece of retinal tissue, which is right at the back of the eye from the front of the eye. So the way that it's done at the minute is by removing the vitreous, which is the gel-like substance in the middle of the eye, and using a long needle to hook the needle under the retinal tissue and deliver the therapeutic. And experience has shown that this is challenging to do in a safe and consistent manner, and often therapeutic leaks out and is lost. So we have access to a technology which was developed initially within Janssen in the context of a cell therapy program, and it's designed specifically to solve this problem, to deliver gene and cell therapy to the back of the eye. And it does it with a flexible microcannula that slides through tissue layers. It's very stable, and a very small microneedle that the surgeon can control and advance into the subretinal space. So this technology is technology that we plan to use in our own clinical programs. We also plan to make it available to partners for their gene and cell therapy programs, and this technology is currently in clinic with our partner, Lineage Cell Therapeutics. In addition to focusing on procedure safety and accuracy, the other aspects of this are thinking about things like consistency and precision. So it's clear that there is a lot of variability patient to patient and surgeon to surgeon in the administration of these kinds of therapeutics. And so a key focus for us in designing this technology has been to be able to really ensure that the desired volume of the therapeutic makes it into the subretinal space. And that it's delivered with precision to the right spot. So surgeon training is a really important part of this. We have an expert surgeon training team as part of our group. And this, of course, all concentrates into scalability. So when we're thinking about an indication like age-related macular degeneration, we need to be able to scale this so that we can apply it globally. So by way of summary, this is an important year for Gyroscope. We have ongoing studies. We're moving our clinical programs forwards, and we're also moving forwards our device development. So we're looking forward to collaborating and working with the gene and cell therapy community as we move forwards. Thanks for your attention.